I'm really here just to give you a closing. I'll try to get in time. And one of the key contributions that Aggie Hirshberg had made, very, very few people know about what she has done. She developed a company called SIDMAP, which is stable isotope-based dynamic metabolic profiling, and being used in cancer metabolomics by NCI, FDA, CDC, and I'll give you some background of how she have developed this company and how impact into the cancer metabolomic that's being used right now in the industry. That's Aggie. She's a visionary. We're very grateful to what she has done to the foundation to us, but very, very few people that she actually started this company. The company is created in Los Angeles. Aggie is the CEO and founder, and his partner are Dr. Boris, Dr. Paul Lee, who has pioneering a lot of the tracer metabolic studies back in UCLA, who is the scientific advisor, and David Manheim, who's the general counsel of the company. Now, SIDMAP provides metabolic profiling services, biomarker discovery, translational research to pharmaceutical company, biotech company, and also academic institution. And currently, over 50+ company and university have used their services. The company was founded in 2002, and I'll give you a background why this company did exist. In 2003, when the Secretary of Health and the Secretary of Agriculture asked me if I would try to put together all the omics for the USDA and the dietary guideline, I was member of that commission. I basically came out with this slide, published in 2003, and this is what we have heard about: the genetic expression DNA, microDNA, proteomics, and down here to the metabolomics. And interaction, nutrient interact, and all of this pathway down here prior to when we are going to go into the clinical studies. And since that time, you could see that the publication in proteomic going up, but if you look at metabolomic, hardly it really exists even around. This is the number of publications that have been looked up by one of my students in PubMed. And it is in 2002 that Aggie has the vision that we need to stimulate to be able to look at the phenotype dealing with metabolism, while these two are moving very, very quickly. Now, by way of background, I just want to get to you that metabolomics is really the study with small molecule metabolites containing cell, tissue, organs, biological fluid, in primary and secondary metabolism. And there are two types. Many people, big companies, are doing metabolite profiling. They're doing basically fingerprinting, but they are not really addressing the metabolic phenotyping. To do this, you have to use a tracer and then try to integrate the input-output to the various biological metabolic pathway. And it is really here that Aggie has contributed in his company on the tracer base using stable isotope. Now, by way of metabolism, this paper here, we all know about the Warburg effect and the effect of how glucose is being handled by the differentiating cells during the oxidative phosphorylation and anaerobic glycolysis. And we know in tumor, in up to what we call the Warburg effect to the aerobic glycolysis. Now, to be able to do these studies, what has been done is that to look at this is where we are doing a lot of the signal transduction pathway going through to the AKT, mTOR pathway and all those things, but we need to link together the glucose metabolism on the other side, and the actor are linked, and using a tracer, we'll be able to look at what's happened in each of these compartments, either in the TCA cycle, either here on protein synthesis, nucleic acid synthesis. You can use the tracer of the glucose. Since there are 6 carbon, you can use any one of those positional tracer to be able to follow the whole metabolic pathway of glucose utilization during normal and also during carcinogenesis and during malignant tissue. So this is the principle behind SIDMAP. In this case, we use 1,2-C13 carbon labeled glucose tracer, which is available commercially. And then you can then follow through using high throughput chromatographic studies, be able to look at each of these compartments, either it's in glycogen, what happened to the little one to pyruvate, lactate, glutamate, or to fatty acid through the acetyl-CoA pathway. So we can follow the tracer. To do this, you can do the heat map. You can create a biomorphic information of the glucose tracer. You have the cell organelle, you have the metabolic pathway. You can follow the tracer, where the tracer goes, and then creating a heat map of where the tracer, you'll be able to quantitate it. Now, it's very difficult to do this prior to this software being developed by SIDMAP, the company that Agi have founded. And by creating their own software, they were able to look at where the level goes and then be able to quantitate the various pathway that you're interested. And to show this an example for you is that there's a new paper that's published by one of the pilot feasibility study done by, supported by Agi Hirshberg, published in Metabolomics from University of Arizona. They were able basically to look at the Warburg effect and the KRAS pancreatic cell. And then you can start looking at the interaction with metformin, and you can see there where metformin affect pathways, where metformin is affecting the whole pathway in that glucose metabolism. So you'll be able to look at that kind of interaction. Another paper published in Metabolomics by Diane Harris, who is now at CDC, surprisingly, all the phytochemical acting at mTOR pathway, AKT mTOR pathway, and yet when you look at glucose metabolism, they're acting at different pathway. Resveratrol is acting here, quercetin here, lutein is acting differently, yet all these three compounds have the same signal transduction pathway alteration. So by doing that, you'll be able to see what happened to the metabolism of the cell and be able to use the tracer approach. There is this is a very, very old paper. When we start playing with this SIDMAP procedure way back in 2002, we were able to look at the phenotype, the metabolic phenotype, and the metabolic pathway we're interested, and we were able to do this. And it was really the background, what Agi then used, the pilot that we have done to establish the company. Now, why the company is very, very important. This was a slide that I stole, given to me by the FDA, talking about drug discovery development. We know where they are right now. You follow about drug targeting. We're talking about personal medicine here, basically looking at primarily using the biomarker or looking at a cellular genetics marker. This is where we go looking at gene expression, biochemistry, following to the metabolic phenotype into the disease process. This is where the drug company are working with, but this is the area of deficiency. If we talk about personalized medicine, we need to understand not only the genetic abnormality and mutation interaction with its environment, and then be able to understand the metabolic phenotype. To do this, we need to do the tracer-based metabolomics. Now, the NIH finally caught up with it. NIH Common Fund put aside $30 million to create a program in 2011. The resource component has now been funded. There are 6 resource centers. Michigan is one of them, Mayo Clinic, and University of California in Davis. And they are now start developing training program to each of these areas, technology development, training program, and this program is available on the website. It's 2 years old. And the concern, I think, right now, what's going to happen to this program during the sequestration situation, whether the program will be compromised. Now, The AACR, you probably have seen the progress report that's being made showing the 4-ring Olympic ring. And I said, actually, there's 1 ring missing. I did talk to the authors that they forgot to add the ring on the environment. They're now highlighting metabolism, energy metabolism, and look at the interaction between what we have done before, moving to energy balance, metabolism, and I think the 5th ring, Olympic ring missing, is the environmental interaction with metabolism. So this is where things are going. So when we talk about personalized health, we are currently basically focusing on gene, protein expression, but we cannot really complete that without not talking about the metabolic index. We need all 3, clinical, we need the bioinformatics to be able to address this issue. So NIH basically said, okay, we have done this, 2 years ago, we need to do this. And this is really a mirror image to what David Wong is talking about salivary, because we are looking biomarker, and not only in each of these areas we have to do biomarkers to all of them to find out what is relevant to the clinical picture that we're looking at. So to you who are interested in using tracer-based metabolomic profiling, it's very important. If you want to use this technology, call Aggie. Call Aggie. If you're an academic institution, it will be very, very basically at cost for you. We have been benefited at UCLA. Our center, our investigator have been benefited from it. But if your industry came out with your dollar and will charge you for the profit, and the profit will go back to support for academia. So the website is there, the telephone number is there. If you want to see what's going on, visit their website is very comprehensive. And I'm sorry I have to do advertisement for you. And there are people in the audience here are currently using the technology that Aggie have supported. So I'd like to thank that we have heard so much of Aggie Hirshberg from Thursday. Some of you have acknowledged during the various paper being presented here the last 3 days. But for me, working with Aggie for the last 10 years, really I have to thank her vision. She's thinking not only on the science, she's thinking about what tool that we scientists need to do, and have a vision to see that metabolomics has been neglected and need to be stimulated. And we're very, very thankful to her for her leadership, her stewardship for our pancreatic research. Thank you very much.